Hwang, a postdoctoral fellow in the Dr. Hwang Computer Research Lab at Weill Cornell Medicine. So today I'm going to share how cytoskeletal control impacts intratumoral T cell lipid metabolism and fate. So as you all know, CD8 T cells are essential for anti-tumor immunity, and when they open activation by dendritic cells in the lymph node, they expand and migrate into the tumor sites where they recognize and kill the cancer cells. And in this process, the metabolic programming and adaptation is important for intratumoral T cell function and fate. However, T cells often fail to sustain their function in the tumor microenvironment because tumor creates a metabolically harsh environment, depriving T cells of key nutrients and inducing the metabolic stress. And further, this disrupts T cell metabolic fitness, leading to T cell dysfunction and poor persistence. And we have particularly interest in this lipid accumulation, especially fatty acid. It's recognized a hallmarker of many tumors, contributing to cancer cell growth, immune evasion, and metastasis. And interestingly, the fatty acid, actually, though capable of supporting T cell metabolism and function, predominantly only fuel tumor progression. So we question, why are T cells losing the fight to fuel their functions in these tumors? So to answer this question, we focus on ovarian cancer as a model system because it's known for its highly immunosuppressive tumor microenvironment, restricted T cell metabolism, and accumulation of dysfunctional T cells, and resistant to all forms of immunotherapy. And I want to highlight this omentum specialized adipose tissues, which play a crucial role in shaping the metabolic landscape of this ovarian cancer. Actually, it releases abundant extracellular free fatty acid that supports tumor growth and survival. And especially these ovarian cancer cells further metastasize into the peritoneal cavity, and these tumor cells further adapt by upregulating the lipid transporters such as CD36 and FABP5 for their and enhancing their fatty acid uptake and the mitochondrial fatty acid oxidation. But we have a very simple question then. How about T cells? So first, we analyzed the lipid uptake in the CD8 T cells in high-grade serous ovarian cancer patient. And as you can see, we observed the T cells from patients' ascites and even the peripheral blood have reduced fatty acid uptake as measured by fatty acid analog, the body P500, mean fluorescence intensity, compared to cancer-free peripheral blood. So these observations prompt us the question, then does impaired fatty acid uptake really contributed to the loss of T cell function in ovarian tumors? Because the other tumor types, such as melanoma and gastric cancer, it has been shown that enhancing fatty acid uptake and the mitochondrial fatty acid oxidation, indeed enhancing the anti-tumor T cell function and immunotherapy. And in this process, the fatty acid transport proteins, such as fatty acid transport proteins, fatty acid binding proteins, and CD36, regulate this process that effectively utilize fatty acid uptake and in the mitochondrial oxidation. So we have very simple question, but very critical question, that how do ovarian tumors disrupt fatty acid uptake in T cells to evade adaptive immune control? So to answer this, first we activated healthy donor CD8 T cells with cell-free ascites supernatant from ovarian cancer patients, and we measured lipid uptake and several transporter expressions. And when exposure to ascites, it downregulated their lipid uptake, and especially we found fatty acid binding protein 5 indeed downregulated when we culture under ascites. However, CD36 and FABP5 levels remain unchanged. So first, we just simply hypothesized maybe we can restoring this FABP5 expression in these T cells may rescue the lipid uptake in these T cells. So to test this, we activated CD8 T cells with overexpressed the FABP5 messenger RNA under ascites culture conditions, and we measured FABP5 expression and the lipid uptake. And as you can see, without ascites, when we overexpress FABP5, indeed they increase the FABP5 expression and accompanied by increase the lipid uptake. However, when we treat ascites, while the FABP5 expression is still elevated, however, the lipid uptake still remain defective. So this raises a very interesting question. Why FABP5 expression, even FABP5 expression is restored, why lipid uptake is still defective? So to make a long story short, we found the T cells from patient ascites in peripheral blood have reduced surface FABP5 expression, but total intracellular FABP5 levels remained comparable. And similarly, when we overexpress FABP5 mRNA without ascites conditions, we saw there's increased surface FABP5 expression. However, when we cultured with ascites, their surface FABP5 expression is not restored in this condition. So this is really interesting so the next question is then how these FABP5 surface trafficking is regulated in this context. So to answer this question, we hypothesize maybe there's a lot of another regulatory proteins that regulate the surface FABP5 localization on T cells. So we use two independent approaches. First is the protein-protein interaction database analysis, and the other is FABP5 immunoprecipitation-based mass spectrometry analysis in CD8 T cells. And interestingly, we observe this Transgelin2 is highly popped up in both of screens. And we also confirmed that by using co-immunoprecipitation assay, Transgelin2 and the FABP5 indeed highly interacting in these CD8 T cells. So what is Transgelin2? Why we focus on this Transgelin2? Actually, Transgelin2 is an actin-binding protein. Actually, it's organized the cytoskeleton and the support T cell function. And it is really important for the T cell and target cell interactions via immunological synapse stabilization. And there's another report suggesting that when Transgelin2 is knocked out in T cells, they actually show reduced cytokine production and the cytotoxic capacity. Indeed, so this means that Transgelin2 is really important for T cell activation and function. And there's another evidence suggesting that this is not a T cells, but in the adipose tissues, when they overexpress Transgelin2 in this adipose tissue, they actually increase the fat accumulations and they expand their adipose tissue. So this is very interesting. So we hypothesize maybe Transgelin2 control fatty acid utilization in CD8 T cells by directing the FABP5. So we confirmed the FABP5 distribution by using fluorescence microscopy, and we saw that using Transgelin2 wild-type T cells, as you can see, Transgelin2 and FABP5 is nicely co-localized near the cell membrane. However, when we using Transgelin2 knockout T cells, FABP5 fails to reach the cell membrane. Instead, they accumulated in the nucleus. So these data are suggesting that Transgelin2 is really essential for localizing the FABP5 on the surface in these T cells. And the next question is then, this Transgelin2 is really regulating the fatty acid uptake in these T cells. So we actually use wild type versus FABP5 knockout T cells, and we overexpress Transgelin2. And we found when FABP5 knockout T cells, they actually reduce the lipid uptake. However, when we overexpress Transgelin2 in FABP5 wild type T cells, they actually promote the lipid uptake. However, when FABP5 knockout T cells, even we overexpress Transgelin2, they cannot rescue the lipid uptake. So suggesting that Transgelin2 is indeed depending on the FABP5 on their lipid uptake. And also, using the lipidomic analysis, we actually found that in the patient ascites, the oleic acid, a major fatty acid in the ovarian tumor microenvironment. And when we treat these ascites with wild-type CD8 T cells, when we cultured with ascites, actually they reduce the oleic acid uptake. However, when we overexpress this Transgelin2, now they rescue the uptake of this oleic acid. So then we have a next question and then what is the functional link between Transgelin2 and these intratumoral T cells in ovarian cancer? So we analyzed the human ovarian cancer patient CD8 T cells profiles, And we found this Transgelin-2 is significantly downregulated in exhausted T cells, which shown all the exhaustion marker. So suggesting that indeed exhausted T cells or dysfunctional T cells, they are highly downregulated in ovarian tumor microenvironments. And indeed, when we analyze our patient sample, suggesting that Transgelin-2 expression is highly downregulated in the ovarian cancer patient CD8 T cells, and indeed, it is correlated with their effector function of interferon gamma expression. And also, we saw the similar phenotype in the mouse ovarian cancer model. As you can see, the effector T cells in ovarian cancer, they significantly downregulate the Transgelin-2 expression. And when we gauge Transgelin-2 low versus high in the same T cells, as you can see, the Transgelin-2 high T cells, they have more interferon gamma production, suggesting that Transgelin-2 is indeed related to a functional aspect in these ovarian tumor infiltrating CD8 T cells. So the next question is, then why Transgelin-2 is suppressed, and what are the mechanisms suppressing this Transgelin-2? So when we analyzed in silico analysis of Transgelin-2 promoter, and we found the several transcription factors related to endoplasmic reticulum stress, like including ATF, ATF4, ATF6, and XBP1 and CHOP, they are highly enriched on this Transgelin-2 promoter region. And it has been known that this endoplasmic reticulum stress is highly suppressed of several immune cell function in the tumor microenvironment. So we have a question that then this ER stress actually blunts Transgelin expression in T cells. So when I treat the tunicamycin, which is the ER stress inducer in CD8 T cells, as you can see, normally activated T cells, they are highly expressed Transgelin-2. However, when we treat tunicamycin, they significantly suppress this Transgelin-2 expression. And to make a long story short, we found the mechanisms that one of the arms of an ER stress pathway, ARE1 XBP1. So this XBP1 indeed directly binds to the Transgelin-2, which blocks the NF-kappa B mediated Transgelin-2 expression in these T cells. So, then based on this link between ER stress and Transgelin-2 suppression, next, our question is then ER stress in driven repression of this Transgelin-2 disrupt the FABP5 mediated the fatty oxidation in these T cells. So first, we observe that when we induce ER stress, we can see marked reduction of FABP5 surface expression in T cells, and followed by reduced lipid uptake. And when we analyze the mitochondrial respiration, as you can see, when we treat the tunicamycin inducing ER stress, they reduce basal respiration and the maximal respiration of mitochondria. Then when we overexpress this Transgelin-2 in these ER stress conditions, we found the lipid uptake is restored and the mitochondrial respiration is rescued when we overexpress this Transgelin-2. So suggesting that Transgelin-2 is really important for rescuing the lipid uptake and the mitochondrial function in these ER stress CD8 T cells. So then, so for final question is that, so based on this knowledge, so can we improve this CAR T cell immunotherapy in this ovarian cancer? So to test this, we utilize the T cells that expressing the chimeric endocrine receptor that use two subunits of follicle stimulating hormone, FSH beta and GC alpha. Actually, it targets the follicle stimulating hormone receptor positive ovarian cancer cells. And this is generously collaborated with Dr. Jose Cornell Garcia in Duke. He's a pioneer of ovarian cancer CAR T cell immunotherapy. And indeed, this CAR T cell construct is now clinical trial. So we are very excited to test this construct. So first, we want to test the therapeutic effects of these CAR T cells in our very highly aggressive ovarian cancer model. And we also confirmed that our ovarian cancer highly expressed this target, the follicle stimulating hormone receptor in the cancer cells. So I injected these CAR T cells in the ovarian tumor-bearing mice, but we couldn't see any therapeutic effects. It actually failed to control this metastatic disease. So, and we found that when we analyzed the phenotype of this transferred CAR T cell, we found there's a bunch of ER stress markers, and they are highly downregulated this Transgelin-2. And when you analyze the Transgelin2 and surface FABP5 expression, they significantly downregulate, which means that these transferred CAR T cells affected a lot of ER stress response that downregulate this Transgelin2 and FABP5 axis. So to tackle these challenges, we generate new Transgelin2 CAR T cells, and we adaptive transfer between normal CAR T cells versus Transgelin2 overexpressing CAR T cells. And we found these Transgelin2 overexpressing CAR T cells actually acquire a central memory phenotype, which we stain by CD44 and 62L, and with increased surface FABP5 expression. And more interestingly, in the omental metastasis region, we saw the better infiltration of these CAR T cells when we overexpress Transgelin2, and we found they produce higher levels of one of the major effector cytokine, interferon gamma. And more impactfully, when Transgelin2 preserves in these SIR T cells, they indeed improve the therapeutic effects of this CAR T cell in this highly aggressive, the ovarian cancer mouse model. So overall, we found that one of the cytoskeletal protein, Transgelin2, which is cooperate with the FABP5, which is one of the major fatty acid binding protein. So it actually helps optimal fatty acid uptake and mitochondrial respiration and antitumor function. And we found this axis is highly suppressed and disrupted by the endoplasmic reticulum stress in ovarian cancer and reinforcing their dysfunctional state of intratumoral CD8 T cells. So based on these findings, we adapt this Beijing knowledge into the CAR T cell therapy. And we found when we overexpress Transgelin2 in ovarian cancer reactive SIR T cells, it increased the surface FABP5 expression and extended intratumoral persistence and enhanced anti-cancer activity. So our findings suggesting that there's another possibility to enhancing the fatty acid metabolism, not only regulating their transporter, but also regulating the unexpected cytoskeletal protein like Transgelin2, so we can find a good way to improve the CAR T cell therapy in any other solid tumors. So I'd like to thank my mentor, Dr. Hwang. He's an amazing mentor, and he gave me a lot of motivation and support. And also, I'd like to thank all our lab members, and there's a lot of great collaborators as well. And I especially thanks to my AACR support for this project and also other grant as well. So thank you for listening, and I'd like to have any questions.